This method should be of interest to researchers who are trying to isolate RNA protein complexes and identify their composition by mass spectrometry. We believe our protocol will be especially useful for purifying complexes that exist in cells in limiting amounts and tend to dissociate during long, multi-step purification procedures.
The main advantage of this method is that it involves only a single purification step and could be used with RNA binding sites of any length and sequence.
Demonstrating some of the steps will be Xiao-Cui, who is a senior research technician in our group.
For binding sites significantly longer than 65 nucleotides, obtain T7-generated RNA and an adapter PCB oligonucleotide as outlined in the text protocol. Mix 20 picomoles of the RNA with 100 picomoles of the adapter oligonucleotide in a 1.5 milliliter tube containing 100 microliters of binding buffer. Place the tube in boiling water and let it incubate for 5 minutes. Then allow the water to cool down to room temperature. First, supplement 1 milliliter of mouse nuclear extract with 80 millimolar EDTA at pH 8 to a final concentration of 10 millimolar. Then add between 5 and 10 picomoles of the RNA substrate that has been tagged with the PCB moiety. Incubate on ice for 5 minutes, making sure to occasionally mix the sample. Next, use a pre-cooled microcentrifuge at 4 degrees Celsius.